John's going to present on the molecular observation network. So John, we'll hand it over to you.
Yeah, great. Thanks so much, Montana. So I'm, first of all, good morning. Welcome, everybody. It's really cool to be here at the summer school. Everyone's been working really hard on the EMSL side. I'm the science area group lead for EMSL's Environmental Transformations and Interactions group. And let's see, I and also Maggie Bowman will be telling you, just providing an introduction to the Molecular Observation Network, which we'll be referring to as MONET. Okay. So the MONET project is focused on the molecular form of soils, at the molecular form of organic matter in soils and its behavior through time and across landscapes. We're focusing on soils and organic matter because they're just incredibly important to understanding climate changes, as well as how we can help mitigate it and sustaining life on Earth. Soils hold a vast amount of carbon, more than in the atmosphere and all the biomass on the planet combined. Another incredibly important point is that all this organic matter is located in direct proximity to the atmosphere. So there's active exchange between soils and the atmosphere. And this means that this carbon is highly vulnerable to climate change. Some of the ways that climate change impacts organic carbon in soils is through atmospheric deposition, changes in below ground biogeochemical cycling. This can be the cessation of certain types of reactions, the acceleration of certain types of reactions, such as mineralization and CO2 release, or methanogenesis. Climate change causes changes in soil microbiomes, and these net out as changes in soil profiles through processes such as aridification, which is important in the West, inundation in the coastal settings, and many others. A really important point is that the molecular form of organic matter mediates how it will react in the environment in response to these changes. So we're really focusing on high resolution form of organic carbon. And the problem is we really have a poor understanding of how this molecular form of organic carbon reacts and the mechanisms that regulate it in the below ground pools across ecosystems. Consequently, we poorly represent these in the Earth systems models. So I'm going to come back to this slide in a minute, but I just want to make one more really important point, which is that if you want to understand how soils across regions are going to respond to climate change, you can't do it by just going and sitting at one field site, making measurements, and then at one point in time and projecting them up and across. This won't work. And the reason why it won't work is because there's so many processes that are occurring at the same time, which are either symbiotically helping each other or they're working against each other. There's also, as you can see in this picture, there's a lot of heterogeneity across the landscape. And you really need to observe. You need to make observations then at these regional scales in order to understand the net processes. And you need to use those then to inform your models. So this regional and higher scale perspective, as well as the molecular organic carbon, this is really an important aspect. And it's a big challenge. How do you even think about the molecular forms of organic matter at regional scales? This is something I think about all the time. And this brings us back to the need that MONET is responding to and the goals, which are really to improve resolution of soil organic carbon and soil responses to climate change, not only locally at the plot scale, but also at the regional scale, and use this information to improve models of soil carbon cycling and storage. Okay, so if you want to understand what we're doing in MONET, there's a few other really critical things about soil processes that you need to be aware of. The first is that biogeochemical and transport events in soils are fast and transient. And this is because of the perturbations that drive them are fast and transient. These include monsoonal rainfall, which is scaled hours. It can be flooding, which is hours to days to weeks. It can be snowmelt, which actually can be very fast. It can be leaf breakout in the spring or senescence in the fall. There are a lot of these processes that drive really rapid biogeochemical so-called hot moments and transport events. And the big problem is that we don't know in advance when these events are going to occur. We generally do, but not really. And the other thing is that conventional single point sampling, on which a lot of the traditional literature has been built, it's labor intensive. You don't know in advance when these transients are going to occur, so you miss them. And you're also sampling in one point in time, one point in space. So you're really undersampling this heterogeneity across time. To do better, we really need new ways of collecting samples. We also need, for instance, sensors, which can record data through time. So sensors are great. They can provide continuous monitoring, but they suffer from low coverage of landscapes. So this is why models are so important. They can help from an experimentalist perspective. This is just my narrow view of the world. Models are really helpful because they can allow you to fill gaps through time and space. But to do this, we need data. We really need this high resolution molecular data that can help address heterogeneity and allow models to really scale across these regional scales. Another big problem is the subsurface is opaque. We can't just look down there and see what's happening, and it's really difficult to access it. It can be done, but it's expensive. It takes a lot of time. There's a lot of promise that geophysical and remote sensing approaches will allow us to be able to infer molecular states and processes that are occurring below ground. But at this point in time, they're really nascent. We really need this molecular data in order to do the ground truthing that we'll be able to do this. So the MONET Strategic Science Initiative is designed to address these grand challenges, and in particular, to address the need for consistent soil ecology and biogeochemistry data across scales, at the field scale, at regional scales, at high throughput, and still to provide high resolution molecular information across these areas. The second big need is to monitor below ground processes through time and across landscapes. This can include, for instance, DOC and nitrogen flushing, microsite activity and so-called hotspot hot moments, nitrogen and denitrification, plant microbe signaling. There's just a lot that we would like to do. All right, so this is a list of the major activities that EMSL believes we will need to be able to deliver to the community in order to say that we've been successful at MONET. The first one, which we're going to talk about the most today, is this automated organic analysis activity. And here the idea is to take the high resolution molecular analysis capabilities at EMSL and to extend them to the high throughput regime and to add data analytics to accelerate this process. In order to get those data, though, you also need these new field sampling methods that can provide improved coordination, centralization, and standardization of sample collection across regions and sensors. This is an important aspect that's been missing. We really need to collect larger numbers of soil samples than traditionally has been done. And this is what's driving the need for the high throughput, both the workflows handling the samples in the lab as well as the molecular analysis. So by doing all this stuff, the automated organic analysis program is going to provide really rich metadata as well as this high resolution organic molecular data, metabolomic, genomic, physical data, et cetera. Maggie Bowman is going to talk about this in just a minute. The other aspects, which we won't talk about today, are really important to be aware of. These are supporting the development of models that can use molecular specific data to improve prediction of organic matter stability and decomposition. The Rhizosphere sensor program and the subsequent field sensor deployment will develop capabilities to detect and utilize chemical signaling to monitor biogeochemical interactions between roots, plants, and microorganisms. And we're partnering with research programs such as the DOE SFAs, Ameriflux, and NEON to collect core samples and to install sensors across intensive field sites and distributed sites as well. So just briefly, the idea then is the goal is to build this molecular observation network, which addresses things from molecular signaling, root soil interactions, and does this at the field scale and the local plot, the regional scale, and across CONUS, which is continental United States. Okay, so our ultimate goal is actually data. It's to create a standardized high-resolution database that can enable the next generation of soil research, both at EMSL and within the user community. But to do so, we really need to improve the way that we're doing the standardized sampling, handling the samples, and then the high throughput molecular analyses. So we need to start somewhere, and the 1000 Soils Pilot Project, led by Emily Graham, was that initial project. It was initiated to develop a workflow for sampling and to demonstrate that this wasn't just crazy. And with that, then I'm going to stop stop sharing my screen and hand the baton over to Maggie Bowman, who's going to tell you about a lot of really cool stuff. If you've been,
John, can you keep sharing your screen? My slides are in your talk.
Yeah, I can keep on rolling. You caught me just the
Yeah.
right time. You can still see all this?
Yep.
All right. Go for it.
Awesome. So I really want to thank John for doing the introduction on Monet. One of the big challenges we had with Monet was how and where do we even start? And that's how we started with the 1000 Soils pilot. And working with Emily Graham, I developed a standardized collection workflow for soil course, where some of you have actually gotten to experience the kits or will be experiencing these kits shortly, where we send you a large Pelican case and a cooler where you collect a standardized set of cores and sensor measurements from the field, and then you ship those back to us overnight. And when we process them, we divide them into two setups, one for a biotic core, which would be the purple workflow, and one that's an abiotic core, or the yellowy orange workflow. And with those, we process the biotic core immediately, and we work into depth stratified layers, working in 10 centimeter sections for splitting into the top, middle, and bottom of these 30 centimeter cores. And for those, we collect a variety of measurements, some of which to help us standardize and participate in other databases like pH and phosphorus and nitrogen, as well as a series of molecular measurements like organic matter extractions, where we can collect the high resolution organic matter chemistry from, as well as we do X-ray tomography, which is a CT scan of the soil cores, which allows us to see core network structure and distinguish between air, water, and soil within these soil cores. And then we have other standardized measurements like texture, which really help us be able to take our database and integrate it with other databases as we move forward. We outsource a suite of measurements to the Oregon State Soil Health Lab and QO Testing Lab. And those again give us those base measurements that are kind of standardized across a lot of databases, as well as we do DNA extractions, and we're working with JGI and GeneWiz to get the metagenomics from the soil. And then all of that funnels into a database that we're hoping to have a forward-facing or a user-facing experience sometime in the next fiscal year, I believe. Next slide. So here's a little video that we love that our communication team put together of our kit kind of unpacking and repacking itself. I just absolutely love this. And so we've had the experience of developing these standardized kits where you're collecting these large scale cores, and it's kind of we send out all of the equipment you need as well as shipping to and from. We can go to the next slide. And then again, from there, we really want to focus on that we're using a standardized processing methods so that everything is being collected in a standardized and logged way, and we're keeping track of all of the metadata regarding the samples from field collection all the way through every step of that process, as well as the metadata required for GeneWiz and JGI, and all of the sample metadata and calibration data from our partners at Oregon State and QO Testing Lab. And then with this, one of our major goals and one of the big things that EMSL can provide that we don't oftentimes have other places is the high resolution organic matter chemistry. And so what we're doing with this is we are extracting organic matter from air dried soils to then be able to turn around and run those samples on the FT-ICRMS, or Fourier transform ion cyclotron resonance mass spectrometry, which allows us to get tens of thousands of peaks, and we are currently using the SciMax 7T with a 2XR, which gives it the resolution near about 14 Tesla. And one of the great parts about that is that we're getting tens of thousands of compound peaks that we then can go through and pick the peaks and then assign their molecular formulas and infer and assign molecular formulas and chemistry for those. And then we can infer data transformations and metabolic pathways, as well as we're doing liquid chromatography mass spectrometry, which Chetan is helping us with this. And this really helps us get the low molecular weight compounds that we might miss using FTI-CRMS. And so our goal is to integrate these two mass spectrometry platforms together to get a more holistic picture of the organic matter composition that's coming out in these water extractions. And then one of the large parts about this project that I think allows us to have a unique perspective is that we are getting these core to core scale for the molecular structure, which allows us to think about soil moisture distributions and model flow through these soils, which really helps integrate the measurements that we're making to a larger scale. And that's being done through X-ray computed tomography, which is being done by Anil and Thomas Vargas at the labs. And this is kind of another high resolution technique that we're providing. And then in addition to that, we are doing, we're currently working on the gosh, why can't it's not connecting in my brain. The words for this just hasn't been enough coffee yet today. The hydraulic property measurements, and those are being done through a suite of off-the-shelf instruments, which are quickly becoming the standard. I think that's the next slide. And we're working with Tom Wietzma and Mark Ruckold, who are scientists at the labs, to help us integrate the hydraulic properties and water retention curves over the saturated through the field capacity and wilting point of soils to understand the moisture retention within these, which really helps us be able to model further. And so while this 3 instrument setup through the meter group is quickly becoming a standardized set of measurements, these will all help integrate into a large database, which is being put together by Yuri Carrillo and Nicole. And again, we're hoping to have an outward facing, but all of this data is expected to be open source, and that's part of the agreement for collecting it, that all the data will be published and open available and allow us to kind of further science by having these open data products. And then again, John kind of hit on this earlier, but the thing that really makes all of this work is that we are working with an established set of networks as well as individual PIs. And for this, we are in our final round of sampling with NEON. And so we will have soil cores collected from all of the NEON sites, which is a really great breadth, as well as we're working with Ameriflux to get, I think they have 44 core sites. And so we're starting that collection partnership, as well as a bunch of Earth system science partnerships, including science focus areas and river corridor projects. What's really great about integrating with these established networks is that it provides a ton of additional and ancillary data for the context of these individual soil cores, as well as it builds these partnerships and allows us to kind of integrate our data models into other data frameworks.
Do you want me to take over here?
Yep, I'll let you take over here.
So in addition to interacting with the large networks, we are working really hard to make sure that these capabilities can be very useful for individual investigators, graduate students, postdocs, other groups. And to do that, the way that we're going to provide these capabilities is to release the first user Monet proposal call later this summer, probably in the September time range. We're working on this right now. And the basic idea is that you saw the soil kit that Maggie just showed, right? So the idea is then we'll have this kit available to send out. There is a standardized set of instructions. You can collect cores of your field sites, send it back into us. This provides data for our database, but in return, you get access to this abundance of high resolution molecular data for organic matter, metabolomic data, genomic data, hydraulic properties, everything that Maggie just talked about is all for free. And we are hoping that this can be a real asset to your research programs. So please think about this. If you're interested, reach out to myself or to Sarah Leichte or to Maggie Bowman. We'd love to chat with you about this. So I'm just going to wrap up. Really our big goal here is this database and being able to share it and make it available to the science community. So over the next few years, we're going to continue to develop our ability to build up this database. To do this, though, of course, we need to develop the standardized sampling, the high resolution, high throughput capabilities. And this means we're going to have to bring online fully this automated organic matter analysis capability. So that's coming up next. We're actively working on it. We'll also deliver modeling capabilities to user programs. The models are super important. If you're not a modeler and you're wondering, why do I need to do this? Well, it's because this massive amount of data that's contained in the database can be encoded into the models, and then you can use the models to understand what processes are occurring when and what the net products are. Okay, so just want to continue to talk about the database here for a minute, because there's some really important things that everyone should understand. First of all, I just want to emphasize the importance of standardization. Maggie did a really nice job of emphasizing how incredibly important it is to standardize from the field to the laboratory when you're making these measurements. So that perseveres, right? All of it perseveres through the entire workflow. So the idea is then that we really minimize non-systematic errors through this standardized approach, and it results in this tremendous amount of data being stored in the database. So there's molecular analysis, the bulk and the elemental chemical analyses, the physical measurements such as CT, this tremendous information from omics analysis, et cetera. We're going to be talking quite a bit about metadata, though, in addition to the data. And that's because the metadata are super important. So what are metadata? Well, metadata, to me at least, this is all the ancillary information from the field, antecedent moisture conditions, the greenness of the site that day, aspects about how you collected the samples. It's also everything that went into the data analyses. These are things that typically get written down in notebooks. And the idea is that by getting the sample metadata, by documenting it and drawing it into the database, we can begin to encode a lot of the domain expertise that went into all the little steps. And it turns out this domain expertise and this metadata can be really important to standardizing the workflow, right? So, for instance, having standardized annotation mechanisms, or to understand peak shapes and interpretations. So this is a big aspect that we're focusing on, is this metadata. The other thing is it's very important for the data to be so-called fully democratized, to be available to everybody who would want to use it, which means it needs to be fair. So it needs to be findable, accessible, interoperable, and reusable. And the point that I just want to make is that all the activities that are going on so-called in the background as we build up this database, capturing, processing, organizing, storing, and sharing, they're all designed from the ground up to provide this fair capability. So this is really the big vision. We've touched on it before. I just want to finish on it. What we want to do at a very high level is to collect and observe high resolution molecular scale information, but we want to do it at regional and larger scales in a systematic way. Why do we want to do this? Because we want to make data available to the science community as fair data. And by doing this, we can help stimulate the use of these data to solve new scientific problems. And arguably, we've never needed this kind of a resource more than we do now with intensifying climate change. And with that, I just want to say thank you. I know we're finishing a bit early. That's because you don't need to take a lot of time to talk about important stuff. And we want to leave time for questions and for breaks. So with that, I'll stop and hand it back over to our MCs.
Thanks, John. Yeah, we had a couple questions come through on the chat as well as on the Discord. One of them from Zuni Kaler, and I apologize if I'm not saying your name correctly. They asked, how did you end up with these measurements at these specific depths? Or is it at all measurements listed at all of the depths? Or is it all of the measurements listed all of the depths?
Oh, you're muted, Maggie.
Figured it would happen, shoot. Yeah, I can address that a lot. We spent more time than when you're talking about it, it doesn't seem like it should take that long to decide on these measurements, but working with Nancy Hess, who retired and left the project, and John's filling in Nancy's kind of hole at EMSL as the group lead. But we spent probably the better part of a year meeting with scientists from all different areas and research to try to identify what elements did EMSL need to bring and what elements can EMSL bring that other people don't have. And that's really the molecular characterization. But then in addition with that, we needed to have a subset of measurements that would help align us with other databases. And so for those, we chose respiration, texture, and general soil chemistry, and anion and cation chemistry to help us align with those additional databases. And with that, we are working with partners that aren't us because they have a standardized method, and we do those on the top and the bottom 10 centimeters of every core, and then we have the middle reserved right now in case we need extra soil mass for other projects. But yeah, so we are doing all of the measurements on the top and the bottom of every core that is 30 centimeters. And where we don't have 30 centimeters, we're doing it on the top and the bottom 10 centimeters, and then we're reserving the middle remnant. So I don't know if that answers the questions.
Yeah, we can keep going. So you addressed this one in Discord, but just in case other people didn't see it, for 1,000 soils, are you doing amplicon and metagenomic sequencing? What are you targeting for metagenomic specific genes or trying to assemble genomes? Thanks for any insight from Becca Evans.
And I'm going to admit, metagenomics are not my area of expertise, and this is handled predominantly by Emily, who is on paternity or maternity leave. But I believe that it is untargeted genome assembly using amplicon methods, and somebody can probably tell me if that makes no sense, but we are not targeting specific genes in this. We're doing a general library for all of the genomes that we're sending out. We're getting the large libraries back of the genomes. We aren't looking for specific targeted genomes.
Thanks, Maggie. From Courtney Price, you mentioned briefly the data will be publicly available. Can you tell us whether any is currently publicly available and which databases it's available across?
The data is, we're working on it. We're getting it. I think we're about 50% of the way towards getting it publicly available. And then it will be published through our database. And then it will probably be linked to other databases and incorporated into like SOTA or other data harmonization efforts through APIs, but we aren't quite there yet.
Leanne, if you're on, do you want to add something to that response? If you happen to be listening and you're within reach of your mute button. Yeah, go for it. Guess not. Okay.
Her video turned on for like a split second.
Yeah,
so.
I thought we were going to get a response.
Okay. Okay, so we have from Miho Yoshika, I think. I apologize again. I'm not great with names. Follow up to Zuni's question, why stop at 30 centimeters and not sample deeper?
Oh, this is actually a somewhat legal question. Anything deeper than 30 centimeters, and because we're a government-funded project, there has to be a staff archaeologist at the site where we're collecting if you're going deeper than 30 centimeters. So that kind of helped determine our maximum depth. Is that anything further requires an additional layer of permitting and a staff archaeologist to be present during sampling.
All right, it looks like John, you just responded to Cassandra Miller in the chat. She asked about information on samples and turnaround time when if you're sending in a sample. Can you speak to that one?
Yeah, right now we do a lot of our processing immediately. And so we collect some of the data very quickly, but then as Josh kind of addressed, is that the hydraulic properties are a bottleneck as well as the metagenomics. We are just now getting our first round of genomic data back from GNOIS, and we're still waiting for some from JGI. So those are kind of the bottlenecks, but I would say it takes about, depending on when your core comes in and when we have these major batches that go out to GNOIS or JGI, anywhere from probably 3 months to 6 months to start getting the full data suite back.
Thanks, Maggie. From Marina Caballero, do you also accept permafrost cores? How does the sampling differ? In the past, we use 1 meter auger cores, so I'm curious how you would standardize the sampling in this case.
Yeah, we are working with Neon to do some permafrost coring, and we definitely would love to add permafrost cores, and we are currently thinking about how to standardize those cores. And I think honestly an auger core is fine for the biological core because you're still getting that depth stratification, but we are losing the structure, which that might have to be just a way in which we acknowledge and accept the limitations of permafrost coring. Neon will be sampling when it's frozen, and they're using an ice drill to kind of drill it out and get us these solid cores sometime in this winter. And then I see Sarah Fisher's question up about if we have samples from heavily contaminated or anthropogenic environments. And yes, we've currently partnered with a CZNet group that's focused out of Baltimore, and they provided us with some anthropogenic city cores from Baltimore. We have a lot of sites from wildfire and other impacted areas that are either experiencing high levels of erosion from mudslides. And so we are accepting cores across all of it. And with the included in the metadata that we're collecting about the samples, we are capturing things about fire history or severity or urban or agricultural amendments.
Great, thanks, Maggie. We have a question in the Discord from Gil. For LCMS data, are you identifying the individual compounds in soils or looking for general compound classes?
It's untargeted. And so we are not targeting on individual compounds. And so we are assigning peaks from the untargeted spectra.
Thanks. Okay. From Monica Schmoll, for metagenomics data, do you analyze both data from bacteria and fungi?
We don't remove any fungal hyphae, and so I would assume that we are capturing some from the fungi, but we are not targeting specifically the fungi, but we aren't removing any of the hyphae or roots that would be associated with fungi.
Thanks. All right, from Vanessa Perillo, would coastal sediments be with microbial mats be able to be analyzed?
Yeah, we've done some sampling along a river corridor transect where we definitely went into some sediments. However, we are not, there are other projects at PNNL and the DOE, like Compass Exchange, which do kind of focus on the sediment to soil connection, and they have a standardized set of sampling similar to ours. And so I think that like Compass Exchange might be a better fit for coastal sediments because that aligns more closely with their vision.
Thanks. All right, and then we have so far one more from Dr. Abisk. What can be the duration between collecting the soil samples and analyzing the metagenome, which I think John touched on in the chat, that it could be somewhere from 3 to 6 months. Do you want to add anything else to that?
Yeah, so the DNA is collected the day we receive the core in. So ideally, cores are collected Monday through Wednesday, and then they're shipped to us on Wednesday. So they're arriving at the lab on Thursday. And then DNA is subsampled on that Thursday and stored at -80 until extraction. And so we are getting ideally under 72 hours from collection to DNA being stored in a safe way. And then we batch extract them and send them out to collaborators, which that takes roughly 3 to 6 months from the time the collaborators receive the sample to the time that we're getting the data back.
Great, thank you. Another question from Miho. Are macrofauna like earthworms and nematodes included in the genomics work?
We do remove any earthworms that we see, and those poor guys get autoclaved. Unfortunately for them, I feel really bad every time. But things that make it through the 4mm sieve that aren't obvious bugs would be included in the DNA work, assuming that they're subsampled and they're in soil in a coarse enough manner.
Great. From Natalie Delherb, would the DNA extraction protocols be available so we can eventually continue to replicate them at a smaller scale?
Yep, and we use a standardized sampling method and I think it's Zymog kit. And so all of that information is collected and will be available when the data is uploaded to the database of how much soil was used, which kit was used, and when it was extracted.
I missed a question earlier from Ido. Will the Monet sampling be limited to the top 30 centimeters, and will it require the use of standardized sampling approach, which was briefly touched on, but we'll bring that up again.
I might let John field that one.
Well, you know, gosh, this is kind of a tricky one because I'm really interested in what's deeper than 30 centimeters, but this has to be a scalable project. And even if we just focus on the top 30 centimeters, we're going to be struggling to handle all the cores that we get. In fact, coming back to this question about soil coring, you know, what we need to do is immediately get collect the DNA and preserve the samples at -80 C, right? But then the drying takes a long time. So we're trying to figure out how we're going to get lots of cores and manage these steps with this relatively small processing team. So right now, the current paradigm is set for the top 30 centimeters, and that's what we're going to focus on. However, you know, we do have a science advisory committee, and we're starting to really dig deeply and understand what are our objectives going to be for the forthcoming years. If I had to guess, I'd say right now we're not about to change the standard process, because the problem is when you add every little thing and then you blow it up over hundreds and thousands of cores, it quickly becomes unscalable. But what we are planning to do is to have individual research campaigns through different seasons, right? So we may have multiple campaigns in a year focused on specific science questions. And you can imagine one of these might be to compare that top 30 centimeters to the next 70 centimeters down. And that will give us an opportunity with a much smaller sample size to target and illuminate answers to certain scientific questions. So we're hoping to do that.
Thanks, John. All right, we have quite a few more questions related to the metagenomics and the DNA. So from Max Colton, do you try to remove relic or extracellular DNA?
We've hit my maximum level of utility on DNA. I'm so sorry.
Yeah, I feel that.
Yeah, I'm also very much hoping to learn a lot tomorrow. And the question the one I can address is, do we have issues with depth
Thank you.
and DNA? And we do oftentimes have to re-extract and concentrate, particularly the lower depths. And so then we're re-extracting and concentrating in order to meet the QA/QC minimum set by the analyzing groups.
Thanks.
And then is there a reason why we went with the Zymo kit versus the Quigen kit? I think that comes down to we're working with a DNA scientist at EMSL, and that's the kit that she said to use. So I'm relying on the expertise. Yeah.
Yeah, great. There was a question about the process for requesting a sample kit, which Maggie already answered, and you can send her an email or send an email to the1000soils@pnnl.gov, and you can apply and get the process started. From Miho, we have, what kind of information do you gather in terms of soil history for each soil core, especially if the ground is not natural or has been manipulated in some way?
Yeah, and so this is we're working with Montana on using the standardized soil metadata sampling sheets, which Montana has put a ton of effort into. And with that, we capture things like fire history, any treatment or alteration history, agriculture. And so there's, I think, like 15 different impacts on soil that are captured within it.
Great, thank you. All right, we've got a couple minutes left. I don't see any additional questions coming through. So with that, I think maybe we'll just take a short 3-minute break. Come back at 10 o'clock to hear from Emily Alo-Fadroche about the National Microbiome Data Collaborative. And again, if you have any additional questions or want to know anything else, check out the Discord. We have individual day channels for questions directly to the day and to the speakers, as well as the lobby for a good place to chat and get to know each other. So we'll see you all again at 10 o'clock.